Okay, let's begin. We have a long session today. Well, good morning, everyone, and welcome. Thanks for joining us today for the webinar on Tai management and restoration activities. My name is Ajay Sharma. I am Assistant Professor of Silviculture at the University of Florida School of Forest, Fisheries, and Geomatic Sciences, and West Florida Research and Education Center at Milton. I'll be moderating this webinar. Please note that this webinar is being recorded. By attending this webinar, you consent to being recorded. However, you will see that videos and mics of all the attendees have been disabled. So the concerns about privacy are minimal. The videos of the speakers are functional, of course. Chat function on Zoom is enabled for everyone, and chat room will be the main medium of communication between all of us, between the speakers and between you and between speakers and the moderator. So please use the chat box to ask any question or make any comment. We have 3 presentations today. After every presentation, I will facilitate question and answers. Also, as you know, this webinar has been approved for 2 education credit units for Florida pesticide applicator training, natural area, weed management, and forestry pest control. If you already indicated in your registration that you would like to receive these credits, we have your information. If you did not, please send us an email to me or to Dr. Pat Minogue in next few days, and we'll follow up on that. Well, we have 4 speakers today. Ms. Megan Keserauskis Kunzer, who is a regional biologist with the Florida Fish and Wildlife Conservation Commission. Ms. Jana Mott, who is project coordinator with the Nature Conservancy and the U.S. Forest Service. Mr. Justin McKeithen from the Forestry program at University of Florida at Milton. And of course, Dr. Pat Minogue, silviculture professor at the University of Florida School of Forest, Fisheries, and Geomatic Sciences, and located at North Florida Research and Education Center at Quincy. Well, I won't take more time. We'll begin the webinar. So first, I will request Megan Keserauskis Kunzer and Jana Mott. Megan served as the project manager, and Jana was the field manager for this project. Both Megan and Jana will provide the background of the project and justification for this more than 2 years of research on Tai control. So Megan and Jana, if you are ready, I'll let you take control.
Thanks, Ajay. Yep, I'm getting to the share screen. I am just actually trying to get to, this is a little bit different than yesterday. Working on, there we go. Okay. All right, Jana, I'm approving you. All right, so Ajay, do we look good?
Yes, it's in a slide mode. So I don't know if you want to do like that, or just maybe you can just show us one major slide at a time, the main slide.
Yeah.
If you go to display settings, maybe there on the top. No. Yes, perfect.
There we go. Excellent, glad to see it. Well, good morning, everyone, and thank you for attending. We hope that there's a lot of useful information on this webinar for you today. I am Megan Keserauskis Kunzer. I am an FWC biologist. I'm housed within the Aquatic Habitat Conservation and Restoration section of FWC, and I focus on ephemeral wetland restoration within the Florida Panhandle. I'm the project manager for this. FWC funded this research. This was brought to us through conversations with numerous land managers throughout the Panhandle. And we appreciate the fact they brought their concerns to us and the need for research. The main partners in this project are represented by the logos below. We are grateful to UF for being willing to take on this challenge for us, and to the US Forest Service and Nature Conservancy. Our project sites are Apalachicola National Forest and Flint Rock WMA. One of my site managers and expertise SMEs is Jana Mott, and I will let her introduce herself. She'll be doing the first half of this presentation, and I'll be doing the second half. And this is just a little bit of background and why we're doing the project, a little bit of justification. Jana.
Hi there. Megan said, I'm Jana Mott. I work for the Nature Conservancy as the North Florida Stewardship Projects Coordinator, for those of you that don't know me. And I also work in partnership with the US Forest Service at the Apalachicola National Forest. And a lot of my work focuses on habitat restoration in these ephemeral wetlands, especially for the frosted flatwood salamander, one of our imperiled species. Our objectives today for this introductory presentation is to give you a background and to define what an ephemeral wetland is, its habitat importance, what our end objective for restoration has been, and the current restoration tools we're using, which led to the justification for the research project that Justin and Pat will be talking about for the bulk of this webinar. Ephemeral wetlands are a niche community embedded in our upland ecosystems in Florida, specifically our longleaf pine historic ecosystems. They go by ephemeral wet ponds, seasonal ponds, temporary ponds, vernal pools, isolated wetlands. We often just start talking about ponds, which isn't really accurate, but it's kind of, you know, the lingo. Seepage streams also qualify as ephemeral wetlands. They're defined by drying up seasonally. They hold water temporarily after rain events and during seasonally wet periods. And because they can be completely isolated, because they're seasonally dry, they are generally free of predatory fish species, which is really important for their makeup and how they function in the larger ecological community. And what's very important is that they are a fire maintained community. They are embedded within our fire maintained longleaf pine habitat, and they are indeed fire maintained themselves. Their habitat importance is very large compared to the size of these little wetlands. They are basically grocery stores for the entire surrounding ecosystem. The biological diversity and biological production that comes from these sites far outweighs the size of them. There's lots of species that rely on them for all or part of their life cycle, including game species, non-game species, you know, larger megafauna. They're the repository of a lot of rare plants. They hold water for birds and wildlife when other parts of the landscape dry up, and they actually have a specifically special importance to amphibians here in Florida. In Florida, about 28 species of amphibian use these wetlands for part of their life cycle, and half of those need these ephemeral wetlands in order to thrive. And of course, our restoration and our focus on them has been driven by imperiled species, including reticulated and frosted flatwood salamanders, Panama City crayfish, Florida bog frogs, in particular. All right, I don't seem to have control over this, so it's not moving for me, Megan. Thank you. So all of those characteristics combine to create a habitat that has 0 to minimum tree canopy, a dense, diverse ground cover, which is especially important to the species that use these sites, and of course the ephemeral hydroperiod. And for this presentation and the research project, we're going to focus on these ephemeral wetlands that are embedded within our longleaf pine flatwoods habitat. That's the work where we've been doing, and that's where tie-ties become a problem that we wanted to address. Here's some more examples of what great ephemeral sites look like embedded in our flatwoods or what flatwoods or savanna habitat. There's an ephemeral wetland on the top left, a seepage stream on the bottom left, and on the right, you can see the complexity of the grass and herbaceous layer that's especially important to some of our pond breeding amphibians like flatwood salamanders, who need the diversity of height and density in order to lay eggs and breed effectively. The habitat's been degraded over decades and decades by the usual suspects: hydrological disturbance, fire exclusion, and inappropriate fire regime. You know, roads, plow lines, etc., draining of our flatwoods for a variety of reasons, including pine plantation, alters the hydro period and makes them less effective for pond breeding amphibians specifically. And it also alters how fire interacts, as does altered fire regimes that we implemented when using prescribed fire exclusively instead of historical wildfires. You know, even in some of our really nice upland habitat, you know, these gorgeous pine flatwoods, we typically tend to burn and apply prescribed fire when our ponds and our wetlands are wet. They're holding a lot of water. And as this niche community hasn't been really to the forefront of our minds for a lot of, you know, the past several decades, it's only been recent, fire's just been excluded. So we have these great flatwoods that have severely degraded isolated wetlands. And then of course, even in our communities where the uplands aren't great, of course, the wetlands aren't as well. And this all combines to create a pond where fire is not effective. And the more degraded it becomes, the less effective fire can be alone to get the pond back to where we would like it. So we've looked to other opportunities for restoration, with the end goal of our restoration being we can get these sites to a point where fire can be the primary tool to maintain them in the required conditions. The types of restoration that we've used have mostly included what we call light mechanical treatment. These severely degraded ponds, they're overgrown with hardwoods, you know, decreasing the herbaceous cover and creating, you know, an increased organic layer. So we go in by hand. We're mindful of our sensitive species and the sensitive habitat, and have crews go in and clear out the hardwood or other undesirable tree and shrub species using chainsaws, possibly brush cutters, you know, cutting them off completely or girdling them and using hack and squirt, and then we treat them with herbicide to prevent or reduce resprouting of these species. We've also, over the past couple of years, started using what we call heavy mechanical treatment, and the photo on the right, where we bring an excavator or a bulldozer in and scrape out. It's a lot more drastic restoration, and we've primarily been using that in areas where we don't have sensitive or imperiled species, and where the peat or duff layer and root mat is of more consequence. But what we are focusing on here is the light mechanical treatment that we've been using herbicide with. And that's what we've been doing over the past decade and what has driven kind of our desire to see this research project come to fruition. Here's just a couple of quick examples of what we've done in the past, so pre and post treatment photos. This is on the Apalachicola National Forest. And you can see 5 years post-treatment, this pond actually responded pretty well. The ground cover responded over time, and we've seen that happen. There's sometimes it doesn't respond terribly well, depending on what is remaining after the trees are gone. But, you know, it's been a mixed bag, but we've certainly seen quite a bit of success. And here's one more example in seepage stream habitat, pre and post treatment. And in this one, you can see the ground cover started to respond fairly quickly. And from there, I'm going to turn it back over to Megan, who's going to talk about the herbicide techniques that we've used in our restoration projects and how that led to the research justification for Pat and Ajay's and Justin's project.
Thanks, Jana. Hello, everyone. Herbicide has been used on many of our projects. It's not a decision that we make lightly, but we have to keep in mind long-term habitat maintenance. And while we want to use fire long-term, we need time. You don't always get fire in immediately. The conditions have to be right and you're trying to be effective. So what herbicide does is it stunts and or kills the target species so that we have time to get fire back in. And that really does depend on ground cover. Ground cover ends up being the fuel that carries the fire across these wetlands. So we make the choice to use the herbicide. But when we make that choice, we know we're working in a sensitive habitat. And so it's all about the right chemical at the right place. On the projects I'm involved in, we use aquatic labeled herbicides and aquatic compatible adjuvants. While there are terrestrial herbicides that can be used, typically it's either in a dry wetland or it can't touch the water. Well, weather is unpredictable. It is Florida, so we err on the side of caution and stick with aquatic labeled. Indicator dye is about precision application. It's about knowing where you're spraying your herbicide and making sure it comes in contact with the right target species while avoiding your non-targets. And also it helps the applicator safety-wise. The precision application that you're going to hear me talk about is about good stewardship in such a sensitive wetland habitat. I'm going to briefly describe 3 herbicide application techniques that we use in our restoration projects. And that's because these are the 3 techniques that you're going to hear about today in the research. The first one is foliar, and we're going to say right off the bat that we know this is the least precise herbicide application technique we have. It involves using a backpack sprayer to spray herbicide on a target species. We focus on shrubs that are under 5 feet in height, but you have to cover the plant, leaf surface, and all that for absorption. We use adjuvants here to help the herbicide stick, so it's less likely to run off. But we know, unlike the picture on your left, when you spray with a backpack sprayer, there's a chance of overspray and putting herbicide on non-target species. So that has to be weighed when the decision is made to use this technique. But if done correctly, you will see the picture like you see on the right. These are not the same areas. They're both on ANF, but they're completely different areas. It just is to show that that's a foliar area that's been completely treated and killed. We are more likely to use what you see here, which is the cut stump application. And this involves felling a tree and painting, treating the cut surface. You could do this with a sprayer, but there would be splashing. So what you typically do on our projects is we paint it on the daub wick paint method. And that's actually what this technician is doing. She's using a metered sprayer to put herbicide on a paintbrush to paint the surface. And with small stumps, you paint the entire surface. And on large stumps, you only have to paint the outside edge, the cambium, the live wood area. And that's what you see on the right-hand side. And that's the beauty of dye, where you can see exactly where that herbicide went. It's precision application. Very little herbicide is going where it's not supposed to be. And finally, we have what you will hear in the other 2 talks as cut stem, but it's also hack and squirt. This is our 3rd technique. This one's not used as much, and you will see it in the photos with the kind of the blue scallop. On the left-hand side, it's a little further out, and on the right-hand side, you're seeing a little bit zoomed in of these cuts that are made all the way around the tree. As this technique is not used that often, I'll go into a little bit more detail. With this technique, you're using a sharp implement like a axe or a machete to create a cup. And these are cuts that go in at about a 45-degree angle, and the depth of the cut needs to get into that live wood. When you create this cup, it's going to hold the herbicide mixture, whatever you're choosing to use, until it's absorbed into the tree. So you're keeping the tree alive, and the herbicide is just entering the transportation system of the tree and being moved around. So you're going to hear more about this. If this technique is of interest to you, I encourage you to contact UF IFAS. Please note that with the cups, all you do is fill the cups with herbicide. You don't need to overflow. I encourage you to read the labels when it comes to this technique, as the number of cuts you need per tree does vary depending on the herbicide you use. Over the past 10 years, we have been doing this work and using these techniques, as Jana has already said. Good stewardship is going out afterwards, regardless of whether a contractor did this work or staff, and seeing how well we did, how effective were we. So these are post-treatment assessments. And while we did this, we realized we were making a lot of really good progress, but we saw that there might be an area where we could use some improvement. On some of our projects, not all, but on some, we were seeing foliar application where stuff got missed. It just wasn't dead, either got missed or wasn't treated well enough. And that's what you see in these photos here is missed vegetation or resprouting vegetation. Same thing with cut stump. You would see on these, this one that's furthest to the left, it almost looks like a chia pet, but there's a stump in there that's resprouting. And that's what you see in all three of these photos is stumps that are resprouting. And the same thing with the cut stem or hack and squirt treatment, where you have trees that they just haven't died yet. They were treated, we know they were, but for whatever reason, they didn't pass, they didn't die. And during our surveys, we noticed that numerous species were represented. But if one had to dominate, it was by far Tai. And this was both Cliftonia and Cyrilla. Just a picture of the Cyrillas here. These are resilient species. They are problematic in wetlands. They are problematic in forestry. Without fire, they take over. They have a significant root mat. They have clonal rootstocks. They're just a very resilient species. And so we were seeing that this was the dominant resprouter on our projects. And so when we decided there was a need for a project, there was no doubt that this needed to be our focal species. So you could look at this photo and say, well, haven't you made progress? And the answer is absolutely. There's no doubt about this. The habitat is by far better because of the work we've done. But if you look at this photo, you see the resprouting stumps. You can also see in this photo that there really is not good ground cover. So our job is to try to get that ground cover reestablished before those resprouting stumps become a mid-story and shaded out again. Question is, how do you do that? Can you have fire go in? Maybe, but there's no ground cover to help carry it. So now we have to go back to herbicide. We have to go back in, spend more time, more money, have more wetland impact, use more herbicide, slower the recovery for the species, trying to buy time to get that ground cover established. This all led land managers to ask the question, because there was no data, are we using the best herbicides in each treatment type, application type? Is it one herbicide? Is it a mix? How important are adjuvants? We're trying to do our job as efficiently and effectively as possible. And there was a little bit of doubt because we're basing it on our field experience because there was no data. These are all valid questions about how can we do the best job the first time around to set it up for fire, to move on to other wetlands, because there's a lot that we need to work on. So these were good questions asked. And finally, what kill rate should we expect? What's reasonable for our long-term management of these wetlands? So we're happy to propose these questions to UF. They accepted the challenge, and the rest of the webinar is going to talk about their research. And with that, I hope we have time for a few questions.
There's been some questions that have come up in the chat that I've been answering as you were talking, Megan.
Thank you.
Hopefully that helps. But
yes, Jana has mostly answered these questions already, except maybe the last one from Joshua. How are the treatments tracked? With GPS and GS application? Well, maybe. Okay, go ahead, Megan, please.
So when we have a project that is underway, we have a defined project area. And we do a lot of oversight on our projects, especially if we use contractors. And so, yes, we do track them with not only GPS, they have to carry GPS on them, but we also have oversight done daily to monitor the work that they're doing through the project. And then, of course, we keep these shapefiles long-term so that we can do a post-assessment afterwards.
And right, and then maybe in the next presentation, Justin probably will talk about it, how the individual root stocks in this project were tagged and monitored using GIS. Like they all were GPS coordinated individual stocks. So, okay, what herbicide certifications do you require for contractors?
That's actually pretty easy to answer. It's a good question. It depends on the land manager, the landowner. And so we follow their requirements. So for the most part, when we're working on state lands, we have to have one license for the crew. When we work on federal lands, it has to be all applicators and mixers for the most part, but it's area specific. Otherwise, we follow the labels for the herbicide. So most of the stuff that we're using, which is what you'll see in the research today, like with Garlon and glyphosate, we require natural areas or forestry certification. With imazapyr, we're being very cautious, and most imazapyr labels have a public water section in it. That's a little vague. And so it says that if you're working in public waters, you should have an aquatics license. And they don't really define what public waters is. And so since we work on public land, we follow the public waters and make sure our applicators have the aquatic license if imazapyr is being used.
Okay, there's one more question. Does the contractor pull the debris out of the wetland into the surrounding area, or just cut and fall into the pond?
I can take that one. For most of the light mechanical projects, we're having the contractors pull the debris out of the wetland. You know, usually there's a good quantity of woody debris, and we want it to be somewhere that we can guarantee that it burns, or that we can dispose of it and get that biomass out of the wetland itself. Cutting it and leaving it in the wetland wouldn't necessarily accomplish our goals. So most of the material, especially the smaller material, we have them drag out and pile up in the uplands or dispose of some other way, depending on the project.
Absolutely. We are starting to play with a little bit of piling within some wetlands. It depends on when fire is going to be used, how it's going to be used, but we are starting to play with, but Jana's correct. Overall, we usually pull the debris out. We never just do a complete fell and leave. But we are playing with the idea of some isolated piles if we think it will help facilitate the next burn.
Yeah, it really depends on fire and what we can do with that amount of material. Because the goal is for it to go away from the wetland. So if it can be burned up, that's great. If we can get it somewhere where it can be burned more easily. But it should be noted that that's pretty much, that's probably what is responsible for most of the cost of these projects, is the labor involved in dragging that stuff out or disposing it somewhere.
There's another good question. How do you decide which methods to use? Heavy mechanical versus light?
I'll take that one. It depends on many factors. One is imperiled species presence. If imperiled species such as our amphibians are utilizing the wetlands for breeding, then at this time, because this technique has not been completely refined, we don't use heavy mechanical, we just do light mechanical. But heavy mechanical, and that, yes, thank you very much. That totally includes rare plants. Heavy mechanical for is kind of the nuclear option, because this is for wetlands that are really fire suppressed. You almost don't even have a mid-story anymore. It's almost a complete canopy, a really thick duff layer. It would be really tough to hand cut and remove all of that debris effectively, and to get, and hand raking organics is hard work. So that's usually how we decide, is the condition of the wetland, what's present within it helps us decide, and also whether we have access and are allowed to use heavy equipment on the site. Can it be used in the, can we move heavy equipment around the uplands? There's a lot of factors that have to be talked about before we bring, whether we can bring excavators into a site.
Okay, maybe one more question. I think Jana answered little bit related question before. What's the total cost of these restoration projects?
The project cost varies depending on the condition of the wetland. It's not cheap work, whether it's light mechanical or heavy mechanical. It can be as low as what we've seen just for initial treatments, not retreats, but initials of, it can be around 8,000 an acre. It can go over 30,000 an acre for some of the heavy mechanicals. It widely depends on how your disposal is, whether they can pile, whether it has to be taken elsewhere on site, taken off site. There's a lot of factors that go into price right now.
Well, we can ask one more question and then we'll go on to the next presentation. We'll have some time at the end of the webinar for more questions. So maybe one more question we can address. So that's a good question. Oh, well, Jana already answered it, I think. Okay. All right, so great. Okay, so let's move on to the next presentation. We'll have some more time at the end of the webinar for more questions. So please keep your questions. Thank you, Megan and Jana, for a nice background and justification for this study. Our next speaker is Justin McKeithen. Justin was key person in implementing the field part of this project, including the installation of the management, tai management treatments, and also data collection over more than 2 years. He will be describing the methods involved in the research and the description of the treatment. So Justin, if you are ready, please go ahead. You might need to unmute yourself, Justin. Right.
There. That's better. As Dr. Sharma explained, I'm Justin McKeithen, Senior Lab Technician here at the West Florida Research and Education Center. And I work with Dr. Sharma, and I've worked many times with Pat Minogue to implement this type of research and field applications all across the panhandle of Florida. And I'll talk just a little bit about our research methods and the application of the herbicides and the sites, and a little bit about what we saw, just general observations over the last couple of years. And then we'll turn it over to Pat after that to talk about the actual results of the research. So Megan and Jana had approached us a couple of years ago, approached Pat Minogue, looking for some answers to these problems that they were having with their herbicides, with just finding the right thing to treat this tai for the long term. What's the best? Are we using the right mixes, the right chemicals? Are we having too much impact? And so Pat came up with this great research idea to kind of explore these things. And they identified two very similar, but very, very different study sites to install study replications there. And these were at Apalachicola National Forest between Bristol and Sumatra, and Flint Rock Wildlife Management Area that TNC owns there south of Tallahassee, near St. Mark's Refuge. Apalachicola site is more of a mesic flatwood that drops off into what we think of as a cypress gum pond, with a pretty good topographical gradient, a couple of feet, several feet actually. And most of the tai trees were right along the edge. They were within 20 feet of the edge, and they were creeping into the upland. The small saplings, they were way out into the upland. Whereas the Flint Rock site was more of an ephemeral low flatwood, more like what we consider a pine savanna or a wet savanna. And once again, the shrubs and tree form tie were more or less around the edges, but the saplings were found in the interior, mixed in with the sawgrass and hypericum, which really was kind of the dominant understory there. And it stayed wet nearly the entire 2-year period that we worked there. It did dry up in the spring, which is pretty typical dry down for those areas.
Excuse me, Justin, if I can interrupt. Sorry. I don't know about others, but I'm not able to see your presentation. Is the share screen active? Can you verify?
It was. Let me try it again.
It was, yeah. It's for past a minute or so. I can see.
Okay. Sorry, everybody.
Yes, I can see now.
Better now? Okay.
All right. Thanks.
What happened there? So, okay, trying to get the slides to advance. So the experimental design of the study was that at both sites and locations, we applied treatments to 10 rootstocks per treatment for 4 different studies. We did 2 foliar studies, a cut stump study, and a cut stem study, also known as hack and squirt. Megan described pretty well earlier. We tried to maintain a buffer between the larger rootstocks. When I talk about the larger rootstocks, I mean the tree and shrub form rootstocks. So, you know, really anywhere from 2 to 5 or 6 meters tall. Those were the larger ones that we treated with cut stump and cut stem treatments. For the smaller saplings that we treated, we tried to maintain at least a 2-meter buffer between the two. And that was for the directed foliar spray. The challenge in that is that, you know, we really wanted to try to find isolated individuals of tie in these areas. And we understand that these things are probably connected via roots, and that we're really may not be treating exactly a single individual. So we just had to really be careful and make our best judgment on what we assumed was a single individual. So the directed foliar studies, which, you know, might also be considered a spot spray, but now we used a very specific technique, a low pressure, low volume technique. And Pat's always referred to it as directed foliar study. It really limits the overall overspray and impact to non-target species. Those subjects that we sprayed were, you know, less than 5 feet tall. We measured height and diameter of those to begin with. And 1 and 2 years after treatment, we remeasured those, made some estimates of crown reduction, the total crown reduction, kind of the herbicide impact of that, the phytotoxicity codes. And we did non-target vegetation assessments within the drip line of those trees. And we'll talk a little bit more about the treatment that we applied and how that impacts potentially what's underneath it. But this was a great concern to Megan, was that, you know, that these foliar applications were having an unneeded impact on the surrounding vegetation, and that they may not have been worth it. So the first foliar study we did was simply just a herbicide study where we compared 3 rates of rodeo or glyphosate. That rodeo was our trade name that we used, trade name chemical. Habitat, also imazapyr, trade chemical. Combinations of those 3, and we compared them really to Garlon 3A, which is the standard chemical used in these wetlands. And of course, we had non-treated controls in comparison. And we added MSO to this, MSO, methylated seed oil surfactant. to all treatments. You'll see in the picture there, our undergrad student, Sarah Hunt. This is sort of a stock photo we made of her actually applying the chemical to this tie sapling. And she's using this directed method, low pressure to treat this thing. And it's really just a light spray as close as you can to the foliage, only treating the foliage. And obviously, you know, you're gonna have some overspray, but it really limits how much of that there is. And we'll talk a little more about the results later. But this is something I've used many times, and it's been very effective. So the second foliar study, we really just wanted to see whether MSO was having an impact or was improving our efficacy of these same chemicals. So we just used a subset of the initial treatments, some with MSO and some without. And one of those, another treatment with only MSO mixed in water. And no herbicide at all, of course, experimental controls. So the second method we used was the cut stump method. You know, and you probably saw in Megan's presentation, you know, just cut completely off at the ground or within, I think it's within 6 inches of the ground, I think was our application directions. And we treated with a similar set of herbicide applications. We measured every rootstock, diameters and heights initially, and of course then we cut them down. Number of live stem diameters, and where it was present, we didn't expect much damage to off-target species, but where it was present, we did make notes on which species might have been impacted. It's a very precise method, so we didn't really expect much, but we took good notes in terms of what we saw there on the site. Like I say, it's a very similar set of treatments that we used here. 3 rates of rodeo, 3 rates of habitat, combinations of all 3 with a non-treated experimental control where we actually cut a frill around the entire tree, but did not treat with herbicide. And a 50% Garlon as the standard treatment compare. You can see Sarah here, she's treating these, and as Megan pointed out, we used a metered sprayer to make sure we only put the specific amount of chemicals on each stump, just as experimental controls. We also used the cut stem method, as always referred to as hack and squirt in forestry. Megan outlined that it's just a single hack, usually depending on the herbicide application. Some labels suggest just a single hack per 2 to 3 inches in diameter. Some labels suggest cutting a frilled hack all the way around the tree, and then applying a specific amount of herbicide within those cuts. We followed the labels, and that's kind of outlined on the next page. So once again, we measured heights and diameters, took some pre-measurements, and at 1 year and 2 years after treatment, we remeasured these. We remeasured number of sprouts, heights, diameters, that type of thing, and impact of critical species, which once again, very targeted application, so we didn't expect a lot of off-site impact. So very similar to the last two slides with herbicide rates. 3 rates of rodeo, 3 rates of habitat, 3 combinations of each, non-treatment controls, all compared to a 50% Garland standard in the overlapping frill around the tree. And then just a single treatment of just frills, cut frills around the tree. You can see in the pictures there, the picture on top is just a single hack, probably one on the other side, given the size of that tree. And at the bottom, you'll see this is the frill cut. Cut all the way around the tree, but no herbicide applied there, I believe. I have to look at the pictures. So some observations about our applications there. I think I mentioned already the Flint Rock site was really wet. And of course, we were applying these treatments in August and September, which is the window that does not affect the activities of the flatwood salamander or frosted salamander or bog frog, which is in the spring. So we had a pretty precise window that we could treat these trees. And that's kind of monsoon season in Florida, so it was very wet. We managed to get the treatments in without getting rained on. And it did concern us some to be walking around in ankle deep water applying herbicides to all of these trees. I'll owe it to the precision of our students, who really were the soldiers who got out there and put the treatments very precisely on the spot, that we didn't have a lot of off-target impact. Some other observations was that, even after 2 years, we didn't see much off-target impact. We did see a lot of resprouting. That's to go with the nature of it. But even the resprouts we saw weren't terribly healthy. Even after 2 years, these were really poor looking sprouts that we saw. The picture in the top center of the screen, that's 2 years later, and those sprouts are maybe a couple of inches tall. I don't know that tie tree will ever return to its former glory, but it's not looking good at this point. So. With that, I guess we can open it up for some questions if there are any, or we can turn it over to Pat and let him take over for the next section, and we'll take questions after, whatever Dr. Sharma wants to do.
Justin, we do have some questions, and thank you very much for a nice presentation. So one question is from Kevin Robertson. What is the tree size range for hack and squirt treatment?
So the tree size range that we focused in on were what amounted to maybe 4 inches and above. And this was from directions from Megan that would typically be the size that they would target with that method. Larger trees that they couldn't easily remove from the wetland. So, where it's acceptable to have standing dead trees, that would be a method that they would use on those larger trees. Of course, if you refer to your silviculture book, it might tell you something a little different. But in this particular situation, that was where they used them for the larger trees. For anything smaller than that, and some larger trees, we use the cut stump method. But we definitely use the cut stem for the larger trees. Is that all?
Okay, great. We actually have some time for a few more questions, if there is any. Okay, so
We got
people
it.
are,
We do have a question that says, would rain have impacted the treatment site or washed the treatment away? And this was a concern that Megan had because of the application period in August and September, was that some of their contractors had had some heavy rains and they were afraid it affected the efficacy. We had treatments applied, or we applied treatments, and we had rainstorms within 2 hours of those, and we did not see any noticeable off-target impact, nor did our results after 2 years really reflect any poor performance of those chemicals. So no, we didn't have that issue, and I'm not saying it's not couldn't be a problem, but we did not. So we did not track the labor effort. We did notice that the hack and squirt method, cut stem, as we referred to it as, is a little more tedious. It's extremely precise and a little less laborious, obviously, than cut stump treatments. But Pat will talk about the results. It seemed to be very effective. The cut stump was a lot more labor, obviously, but it was kind of foolproof. You just cut it and treat it. I can't say specifically that there was a downside to the Garland application, but Megan did have concerns that it wasn't maybe the best option in every situation. And that's something that while we explored the imazapyr and glyphosate, Pat came up with this wonderful idea. So we just kind of went from there with it. We did not use red oil. We used MSO. That was just sort of the standard.
okay, there is another question. Maybe Megan and Jana can answer it.
Yes.
If you don't remove the dead trees, is there a noticeable effect on subsequent fire behavior?
We haven't been able to track that effectively because we still struggle to get fire into the sites after treatment. And the problem with not removing the trees is that we have heavier fuels. So the effects on subsequent fire behavior tend to be, heavier fuels don't burn as well. So we're just not consuming them when we're trying to get fire into the ponds, into the wetlands themselves. If we happen to get fire in there when it's dry enough that the heavy fuels are actually available, we end up with a little more intense fire, which in some of these sites is not a bad thing. And that's great, but our primary concern is getting fire, and we want to have a range of conditions available to get fire so that we can do it safely, but also while the ponds are starting to dry down. And having the heavy fuels impacting the fuel loading in the wetland hasn't been conducive to that so far.
Right. Thank you, Jana. Craig Iverson says he can't wait to see the results. So I guess we should move on to the next speaker then. Well, thank you, Justin. Thank you, Jana. Our next speaker is Dr. Pat Minogue. Dr. Minogue is PI, Principal Investigator of this project, and the key person behind the conceptualization and design of this research and overall direction of this project. He will be presenting the detailed results of the research projects and the recommendations and implications for managing tai-tai. So Dr. Minogue, if you are ready, I will let you take the charge.
Good morning, everybody. I've been looking forward to this for a while. We've got a great crowd today. A lot of folks, I recognize a lot of names here, a lot of folks that are doing restoration ecology. And a lot of foresters, you know, Titi is not an invasive plant, but it sure is a problem. Yeah, recently they've been calling it a nuisance. I think it's more than a nuisance to me. It has been. Been growing trees in this area since the early 80s, and we've been dealing with Titi. And in forestry, we have pretty well figured that out. And combinations of herbicide and mechanical, but it's not particularly selective. And we also are putting out often pine trees, which grow very fast. So when Megan and others, really Megan taking the lead on that, said, hey, you know, can you help us with this? And I said, I think it's very interesting because we haven't figured this out. We need to find treatments that are selective. And, you know, as I was thinking about it, the most important thing is selectivity by placement, you know, by putting the herbicide on the target. And I also drew on a lot of experience, tens of thousands of acres over, is that 40 years plus, where we have been very effective in controlling this species. And what was really clear to us early on was, you know, we're using Garlon. That was one of the few products we had in the early days, and we did a lot with Roundup. Roundup was really very variable as a foliar spray. Sometimes it worked great, other times it didn't. Of course, it has very limited absorption in through the cuticle. And Garlon looked great right off. I mean, I like it. You know, I look at it about 2 months. Oh, we killed everything. Looks great. Everything's brown, right? Well, it wasn't brown. You know, it was just hiding. You know, we kind of mowed it off on the top, and then it all resprouted. And we brought in mechanical, but really it was the herbicide that solved the problem. And it's about translocation, getting to the root of the problem. That's what it's all about. But I'm going to start by saying this was a lot of work. This was a lot. There were 7 different replicated studies for 10 reps, the way it was designed, and everybody had a part in this. And I want to thank everybody for the hard work they did, all the graduate students that were involved, folks that aren't mentioned here. Justin mentioned quite a number of them. I also always want to acknowledge the funding that we've gotten, FWC, with the Fish and Wildlife Conservation, Aquatic Habitat Conservation and Restoration section, and also with Sam Wan's group, the Invasive Plant Management section, which they were involved with this, even though it was not an invasive, it's of interest to a lot of people dealing with difficult species. So I'm going to page down. Oh, great. That doesn't work. Well, I'll tell you what. So maybe I can do something with this. Yeah, I can use it with my cursor. All right. So I've already talked about getting to the root of the problem. We've talked about the botany. I want to emphasize that this is a root sprouter, and it has a root mat, which is very persistent. And a lot of the individual stems are coming up from sprouts. I've looked into the literature to see what kind of seed viability is present in this species, and I find no reference of that. I think it would be an interesting study. A lot of plants that make a lot of seeds have lower viability. And like many other species that are a problem because of their root mat, we need to really select selective herbicides that will translocate. Imazapyr is a very good translocator. It moves in both the xylem and the phloem, and it moves very quickly through a plant. Glyphosate also is a super translocator, and in particular among all the herbicides that we use in woody plant control, it moves preferentially to the roots, to the root stems. So I wanted to use that in the design, even though the past experience was very variable. It would work sometimes, maybe 25% of the time it worked, the rest of the time did not work. And I thought, you know, we've been using MSO, methylated seed oil. Justin talked about that. And that really improves the control with glyphosate. Glyphosate is not readily absorbed by that thick cuticle on these leaves, waxy species. And it works largely by just increasing the time that it sits. The spray will stay on the foliage. You know, that oil doesn't evaporate very quickly. And it also solubilizes the cuticle a little bit, the cutin, suberin, and wax of the leaf so that we get more absorption into the vascular system and movement down into the roots. Timing, that was another thing that was important because, as Justin said, we wanted to do this at a time that we would not interfere with mating reproduction with our amphibians of interest, the flatwood salamander in particular. So we wanted to be out of the field, out of the study by the end of September. But that fit well because that, you know, at the end of the growing season, those herbicides move preferentially from the leaf into the stem and down into the roots. It's basically an amino acid analog. And in fact, that they work on the production of those buildings of proteins. And as the plant is trying to bring back those proteins and those nitrogen containing molecules back into the system, you know, they're trying to recycle the nitrogen, get it down into the woody plants. And that's particularly true for those that are decidual. Well, deciduous, this is of course an evergreen, but in any case, they will translocate to the root very quickly or well during the late season. So that was the reason that I brought that into the test, and I thought, well, you know, imazapyr is a super brush control, but it has long persistent and it's very broad spectrum and it doesn't have very good selectivity. Yeah, we're going to put it out with very careful placement, but it's on the site. So I wanted to try the smallest amount of habitat, imazapyr, to get an effect, and bumped up the glyphosate rate where we could look at a very range of the labeled rates, and within the label basically went from the low end to the top end. So we could look at rate response, and that's what we're going to look through right now. I'm going to see if I can page down. Oh, it's working now. Good. Well, Justin did a great job talking about the sites from an ecologist standpoint, and he is a good ecologist and really good with his taxonom and very good with plant identification. And as a forester, though, what I see is that, you know, one of the sites, the Apalachicola National Forest, you know, we're doing a lot of intensive prescribed burning on those sites, and you can see where the burns stop and where the tie begins. So, you know, fire is going to be a very important tool as we manage this. And one of the reasons there's so much tie on the Apalachicola National Forest is that first grazing was taken off, and also for many years there wasn't as of an aggressive prescribed burning program. But I've looked at aerial images of that forest, and it's amazing to look at the change over the last 60, 70 years in the amount of tie on that 500,000 acre area, almost that large. So, oops, that didn't work out too well. Let me try this. All right. So the other site, definitely more hydric. You know, it was, I think it's maybe not so ephemeral. Ephemeral. It's really kind of like a pond, at least when we were working there. It was very wet. We didn't have as many tall stems here, so we couldn't do the cut stem study on this particular study. Again, Justin did a great job talking about the sites. And he also went into the treatments. Just to bring that forward, I'm going to start by talking about the shrub form study that we did with foliar applications. And these were stems that were less than 5 feet tall. And the study was basically a rate study. You know, we're going to look at the response for a wide range of herbicide rates, rodeo alone, habitat alone. And then we wanted to look at the proportion of habitat in rodeo mixes. So for this, I picked the middle rate of rodeo, 7.5%. That's a high rate. That's a pretty strong concentration. And then I looked at that over a very wide range of rates of habitat in combination. And then the objective really was to compare it to the operational standard, which is 4% of Garlon 3A. That's the amine. That's the form that is very effective with translocation within the xylem. It is much more soluble than the ester and is very poorly absorbed through the foliage. For foliar treatments, we typically would use the ester. Why wouldn't we use the ester? Well, the ester doesn't have the same selectivity and it has some volatility problems. Also, the fact that we're working with aquatically labeled herbicide. So that was very important that we worked with the 3A. But in any case, that's been the standard. So we're going to compare in all of these studies, it's pretty much the same kind of design. I'll point out any differences that we have. So all of these in this study had MSO at 1%. And that amount will make an emulsion in the solution. It makes it kind of a whitey, almost a milky emulsion. It's not really a true solution. And that improves the absorption by reducing the evaporation rate, okay? You get more residual on the surface. Okay, let's go right to the results. The way I'm gonna do this is I'm gonna give you the answers first, and then I'm gonna explain how we figured this out, and we're gonna pick it apart, and I'll try to give a proof, if you would, for all of these findings as we go through it. And we're going to just take it one thing at a time. First of all, for the directed foliar studies, rodeo clearly provided the best control at 2 years with no difference among the rates. Between 5 and 10% solution, no differences. And with an average over all of those rates, and since statistically we're saying they're not different, we can average them. And then what did we get? Well, 79%. That was our best treatment. And that compares to 4% with the non-treated, no herbicides involved. And height control was 91%, again, our best treatment. And that compares to minus 17% for non-treated. Uh-oh, how do you get negative control? Well, this was a measure of the growth in height from pre-treatment to the measures taken at 2 years. So if we look at that, the reduction in height, a negative value means that the stems were increasing in height. We saw that there was great compatibility with rodeo and habitat, and that is expected. That's what we know happens. They're both fairly slow in their activity. As I mentioned, they translocate very well, and they both work on amino acid synthesis, the building blocks to protein. So they're very compatible and widely used for many uses, particularly woody plant control. And an important finding, which surprised me, it really surprised me, is that habitat did not improve control over rodeo alone. Now, part of that is, as I mentioned, I used very high rates. We used very high rates of rodeo. But even then, even with the higher rates of habitat, it really didn't improve the level of control. But we still, you know, looking at this, we're 79%, 91%. So it's not completely effective. We're still, you know, there's still some live rootstocks. And Garlon 3A increased tie mortality and height control compared to the control, but it ranked below the level control by the rodeo. So rodeo was doing a lot better than Garlon 3A. It's important to note that the effectiveness increased with rootstock, decreased with increasing rootstock height. So the taller stems were more tolerant, harder to control, which that makes sense, I believe. All right, let's pick it apart a little bit. And the first thing I want to do is make some comparisons to the rate effects that we had. And this is an analysis, and all of the analysis that I'm giving you are across both sites, okay? And they're going to be 2-year results. And I think that is really the gold standard for woody plant control. We don't know what we've got at 60 days. In fact, it often is the wrong view. We see things that are browned up, but they didn't get translocation. You know, we mowed the top, and my experience is that slow kill is a good thing. You know, because you want it to get it into the root mat. That's the target. So we did some ANOVA, looking at analysis of variance, and we looked at the effect of rodeo alone, habitat alone, and habitat rate in the mid rate of rodeo, and that was not significant. So there was no rate effects at all. And all treatments gave better tie control than the non-treated control, except one. Habitat did not work as well and was not different, particularly for mortality, from the non-treated control. And as I mentioned, that surprised me. You know, 3% habitat, that's a 2-pound acid equivalent gallon, you know, 2 pounds AE per gallon, 3% of that. Typically, I'm using a 2% with a 2-pound rate for foliar and getting very high control, and just didn't work that way. So that makes me want to reevaluate what I'm doing with some of my forestry site prep too. We're going to go down and take a look at, now let's compare the treatments to our standard. We did some analysis of variance to look at Garlon 3A and looked at the comparison between rodeo and Garlon 3A for mortality. It was different. Height control, that was a significant difference there. And then habitat versus Garlon 3, highly significant, and because habitat didn't work as well. So looking at the averages then, because there is no rate response, we can compare the averages across the products to compare rodeo, habitat, and the tank mix. And the thing that really came out was that habitat was not as good. So I think we're starting to put a pretty strong argument to this case here that habitat wasn't as effective. And And as I say here, the average habitat tie mortality and height control was less than the 4% Garland standard. I think that pretty well covers that. And then for comparison, you can look at what we had the control with the standard, you know. So rodeo was doing a lot better. And yeah, okay, so the tank mix is right there, you know, but the habitat isn't adding anything in there. And there's no reason to make that tank mix because it doesn't add anything. It just adds some residual and less selectivity. Okay, you know, Megan was saying, you need to show me why you want to use MSO. And so, oh, we figured that out a long time ago. You got to put it in. You know, we use that for Kogon grass. We use it for all the really big problem species that we're dealing with, the root mat, the rhizomes. But yeah, we'll put something together. It's a little study. We replicated this on both sites. And this was a simple comparison looking at the middle rate, the middle of rodeo, the middle rate of habitat, and then the middle rodeo and the middle habitat rate with and without MSO. And then we used MSO only, which I think we need to do that because that's your experimental control. And as what we'll see in a little bit, the MSO just by itself with 1% was given a little bit of control. And of course, we always compare it to the non-treated. Okay, by analysis of variance, the MSO generally improved control except for rodeo and habitat in a tank mix. And this interaction was positive at both of the sites. So there's something with this. And in fact, if we look at means here across the two sites, we see in the yellow here that the mortality is actually less with an MSO, and height control, not significantly different, but not as good, I'll call it, with that combination. But statistically, there was a difference, and a difference between with and without habitat. Now, why that is true, I don't know. I've been thinking about it a lot, and you know, thinking about the functional groups in those two molecules and the effect of the pHD of the acidity pH of the MSO in that combination, and protonation of the functional groups. I think that it may explain what's happening there, but that's just a theory, something we could look at. But what's important is we see it clearly. So something to think about if you're going to do a combination. However, as we go through all of this, we're going to see that there wasn't really a reason to do a tank mix. So I guess that's academic, isn't it? I would point out that we're always comparing to an experimental control. So with mortality, the important comparison was MSO alone, which provided 15% mortality. I was surprised by that. It's not known to be that phytotoxic, but it did by itself provide some mortality. And then with height control, no real reason to compare that, but rather to the non-treated control, which again, you see, that's a negative value. So there the control grew in height. Okay, we're going to go to another one now. We're going to look at cut stumps, and that's a mainstay. You know, that's the most widely used treatment in this particular effort. And in this design, very similar. Look at wide rate of rodeo rates all the way up to 100%, and that's the range that's right on the label. Habitat, likewise. In all of these designs, I kept within the range of the labeled rates. And even with the cut stem that we're going to get to, I used the recommendations where we were using the same approach to cutting the stem with overlapping cuts. For example, a Mazapure, you could do wide spacings at a lower concentration, but I didn't want to confound the study. I just used the more dilute solution and kept the same approach. All right. Then again, we've looked at this proportion. Rather than looking at rodeo at just the middle rate, we went with high rodeo, and low habitat to high habitat, kind of a titration to look at that rate response. And we'll look at that to figure out what ratio do you want in the mix. But I'll tell you what we know from most of the work that's done in woody plants is that typically we're using a small amount of Mazapure habitat with a larger amount of rodeo. Part of that is because of the cost, and that's less important than it used to be, but habitat is more expensive than the rodeo. Used to be a lot different in the cost. And again, we're back to the 50% Garlon 3A standard, and then cutting only. I think that's pretty interesting with this species. Let's look at that. Look what happens when you just cut the stems. And of course, this is research, so we always have a non-treated control. All right. We're going to look at 2-year after treatment mortality. And to me, that's when you start to make decisions about what works and what doesn't. The first year, you get an idea. In fact, I was surprised the first year was more similar to what we see in the second than in most studies. But what we saw very clearly is that mortality decreased with increasing number of stems per rootstock, which makes sense. A larger stem is going to take a higher dose. And we saw this was a significant covariate explaining variation in mortality. And because of that significance and the importance of the number of stems per rootstock, Dwight Lauer, who's an outstanding statistician working with weed science and ecology and forestry for many, many years, a colleague of mine, he worked out some regressions to look at the predicted mortality based on the number of stems per rootstock. And what we did was we made the mean comparisons for the average number of stems per rootstock, which was 3.4%. So it's not a round number, but that was in the middle of the pack, if you would. And it was also interesting in this study to see that cutting only caused no mortality. And because of that, we took it out of the stats because we were really more interested in comparing these treatments here. And we had the non-treated in that comparison. So, that was excluded from the analysis because it just would make the analysis cleaner. The bottom line was that tie-tie rootstock mortality was greater than the non-treated control, and there was no difference among these treatments, but Garlon 3A gave 60% mortality. And that was not, as I said, not different than 48% for rodeo, 42% for the high rate of habitat, or the combination of rodeo and habitat. The average tank mixes there. And again, there was no habitat rate response in that. So that you really want to compare it to the high rate rodeo. So I have to step aside right now and think about, so when we are thinking about performance in standard performance for contracts when we're asking for 90% control. We have to think, well, what does control mean? Let's think about that. We're going to look at a lot of different variables. I've shown, you know, so far we've looked at mortality, and we've looked at we'll look at basal area and other variables that we measured, and you'll see across that you know, if you really are talking, what is control? You know, is it the way it looks, or is it the effect that it's having on root sprouting and managing the root mat and getting control and ultimately getting the job done? It's note too that mortality increased linearly with rodeo rate, but the rodeo average mortality was less effective than Garlon 3A. So at the end of this, I'm going to come with a very short list of what works best. All right. Let's look at 2-year resprouts and the tallest height of the resprouts. Now, cutting only increased the root stalk height and the number of sprouts. So by cutting these rootstocks, you're going to increase the number of sprouts. We knew that. We knew that. We see that. This species is a sprouter. But what was really curious to me is look what it did. Now this is the sum. No, this is the tallest stem in the rootstock. So by cutting it, you made that rootstock taller, which is not what we're trying to do. So I thought that was interesting. Back to Garlon, 50% 3A, significantly greater resprout control than the other herbicides, and it was better, highly significantly better than rodeo, highly significantly greater than habitat for the averages of those actives. And when you look at Garlon 3, it was the only herbicide that had fewer sprouts than the cut-only experimental control. Okay, we're going to look at the predicted basal area for our average number of stems per rootstock. And I compare the basal area here, and I've got some mean comparisons in this column, and then percent control. Percent control, which was a change in basal area from initial to 2 years. Okay, so it's an actual measurement. It's not just an estimate or an eyeball estimate. It decreased with increasing number of stems per rootstock significantly. All right, we get it. Larger stems, larger rootstocks, harder to control. Basal control was calculated as the percentage of the cut-only treatment. Okay, basal control was calculated as a percentage of the cut-only treatment. And that's important because cutting increased sprouting, so it had an effect on basal area. All herbicide treatments except for the low rate of rodeo reduced the 2-year tie basal area compared to the non-treated. So all of these treatments except for that 50% rodeo was better than the non-treated. And the average rodeo alone had a greater basal area than Garlon 3A. So rodeo on an average was better than Garlon 3A standard. We saw there was no rate response for rodeo, so you might as well say 5% solution of rodeo was better than Garlon. We'll come back to that. So again, Garlon 3A ranked best, and with basal control, 87%. It was interesting too. I kind of played with some of these numbers. Cutting only reduced basal area by 93%, but increased the sprouts by 1,000%. And that turned out to be an exactly even number, which was kind of funny. All right, we're going to go to the last study that we did, and that was the individual stem treatments. This is, in my opinion, in restoration activity. I'm doing a lot in particular with longleaf restoration. We've done a lot with invasive plants over the years. But the thing I like about the individual stem treatments is that you really get the herbicide right into the translocation system, you know, and particularly when you're going after the root mat. The other thing I really like about this is it uses a very small amount of herbicide. And where, you know, Megan made a comment, and it's so true, when we do foliar spraying, particularly, if, you know, if we're spraying on some tall stem, well, a lot of times we try to keep things less than about 4 feet, maybe 5 feet tall, but we know we go higher than that. And if we go anything higher than about waist high, we're getting other stuff. So it's just not by placement, you're not getting the selectivity with foliar spray. And I really like cut stem treatments. And yes, it leaves dead wood. I think, you know, the sites are going to look as pretty, you know, but through times things melt down, and that wood has some value for wildlife. Holly Ober and I have a publication on dead wood is good. So there's a lot of value to having standing stems, and frankly, just thinking about the cost, you know, this is so easy to do. It's so much easier to go out there with a hatchet, and you could do wide spacing with a Mazapir, you know, unlike rodeo. That would be a reason to actually do some more testing to look at the wide spacing treatment with a Mazapir, because the labor is so much less and it's so much faster. So you could, and for more money and less time, you could do more acres. Something to think about. That's my perspective. I just want to share that with you. Now in this, we had the exact same treatment, so no need to go over that again. Here, what was interesting is that this worked best, you know, cut stem treatments uses less herbicide, and all the herbicides provided 80 to 100% rootstock mortality. Look at, you know, 100% mortality with quite a number of these. I mean, they were all dead, all of them, to get that grade, right? And also looking at the basal area. Here you have the initial basal area. This is before treatment, initial or baseline, if you would. And then after 2 years, the basal area that we had, non-treated control, you can see, you know, it didn't grow up a little bit, but there was a little bit basal area control. So that's an important experimental control. And, but there again, all of these treatments, it's not significant different, but there was, you know, a little more resprouting and more basal area with the Garlon 3A. The other thing to finish up on this is that the cut stem treatments did not stimulate rootstock sprouting, as did the cut stump. All right, that's something to think about, you know. So it's not going to cause resprouting. All right, now Garlon 3A. I'm going to wear this out, but Garlon 3A provided the poorest rootstock mortality, 80% to 97% for the rodeo and the rodeo plus habitat. Again, no reason for the tank mix, but that I think is important. And also, again, no advantage for the tank mix. Oops. All right. and talk about critical species. And this is a very important objective of this work. And I appreciate the work that all the graduate student and Justin did on this with Ajay's team. It was a lot of work. We did assessments at 1 year and 2 year. We typically do a baseline, but we were trying to minimize the cost for the agency. And this kind of work takes a lot of time. It really is a very intensive effort. I need to tell you that the 2 studies, we could only evaluate the results at the National Forest because we had Hurricane Michael roll in about 2 months after we did the treatment at TNC, and things, when we treated, there was still inundation on the site. It was very, very wet. There were places we were walking up to waist deep. And for that reason, we really didn't have a test to look at. And it's kind of hard to get at this, but we were able to get good data from the directed foliar study. Now, why is that? Well, because the directed foliar has more impact. That's a part of it. But anyway, well, let's just take a look at some of the important findings that we had at 2 years. About half of the plots with critical species, these are plants that are important to pond breeding amphibians, had reduction in critical grass species, as wiregrass and these other species that you see there. We had a list that we were looking for. And there was some recovery of critical grasses, in particular wiregrass and these others, with a couple of treatments, the rodeo 10%, habitat 1%, and the combination of rodeo plus habitat. Rodeo is really effective glyphosate on gramineae, on grasses. So that's something that I have to wonder, I guess there's recovery there because the initial effect was a great disturbance to the grasses on the site. But then again, I think when you look at restoration from ecological objective, that it's about time. It's all about time. And we tend to, I guess it's our human nature. We want to see a result right away, but nature doesn't work that way. Things recover. And disturbance, fire. We talk about, fire is really great. It's a wonderful tool for disturbance, and I agree with that 100%, but it's not particularly selective. And so that's something to think about there, that sometimes even these really intensive treatments, we did a study in longleaf restoration, which we're publishing in Ecology, Forestry Management. It showed that with broadcast of the imazapyr, we nuked it. I hate to use that non-technical term, but because of that disturbance with time, it came back to the desired species better than other treatments. So we have to think about the longer term effect. I just want to make that point. That's my opinion. I think it's an important consideration. Now, in terms of the forbs on the site, there was reduction in several that are on the list, and this was noted for the low rate of rodeo plus habitat tank mix. But I think that really, just had to do with prevalence, whether it would happen to be randomly on that particular treatment or not. It's difficult to get these kinds of data with relatively small plots, which we had. The important take home is that Garlon 3A showed the least reduction of total ground cover and critical grass and forb species for the directed foliar treatments. Let's look at the data that we have for this one study, which was the foliar rate, habitat, and proportion in rodeo tank mixes. These bar graphs are paired. You see there are 4 bars, right? The first two are percent total ground cover at I'm just going to get my pointer up here. There it is. At 1 year and 2 year, the first two bars. And then the percentage reduction in critical grasses at 1 year and 2 year are the next two bars. It's interesting with the non-treated, we didn't have any observations of the critical grass species. Maybe it needed more disturbance. I don't know. But that's not a big difference, just little observation. But this clearly shows that Garland was quite a bit different than the other. And rodeo, we're seeing that cover is generally reducing with increasing rodeo. We haven't done rate response, looking at statistically. We have a PhD student that's working with this data, and we're going to have that by the final report this June. We're doing more work with this. I just wanted to give you an update of where we are with these data. With that, let's look at the bottom line. What we saw was that 5% rodeo, maybe less. We didn't look at less, but there was, it was starting to drop off at 5%. Typically, I'm using a lot of 4% rodeo with woody plants as a directed treatment. But again, I would caution that it's very broad spectrum with foliar treatments. You're going to have collateral damage to the associated species. And we got up to 80% tie rootstock mortality and 90% live height reduction. For cut stump, the standard 50% Garland 3A gave best result, not different than some of the others as we saw, but it was the leader. 6% rootstock mortality, 87% basal area reduction. So, just something, think about that in terms of expectations that you're going to have at 2 years. At 2 years, you're going to know what you got from herbicide treatment to woody plants. For cut stem, again, that's the one I like the best. Garland 3 provided poorest rootstock mortality compared to 97% for the average for rodeo and the tank mixes. Again, there was no real statistically difference there, but it was a different, I think. And we looked at 50% rodeo for best safety. Oh my gosh, there's a mistake here. Yeah, it should be right here. Oops, I'm gonna go back to that. My apologies. Need to note that it says 50%, that should be Garland 3A gave the best selectivity. Okay, so there's trade-offs in all of this, there was a really good question. Why do you pick foliar over other things? And Megan had it right. It depends on what the site's like. The size of the stem is hugely important. And typically it's a mix, you sending in some folks have got backpacks that they've got their Garland, that they're going to do the cut stump, and then there's some places where you've got multiple stems so close that you want to go to foliar, and other places where they're taller and you're going in to cut stems. So it's finesse, really looking at that, what's going to work best. But I think that's important to say that Garland 3A did show the least reduction of the total ground cover and critical grass and forb species, but poor control with foliar and cut stem treatment. So that's what I had for you today. And now the really interesting point would be time for some questions. I will remind everybody that we do have CEUs, pesticide CEUs, 2 credits, either for the natural weed management or natural area weed management or forestry pest control is and or. You get, there's 2 credits. You can get 1 of each, or you can get 2 in one or the other area. But if you registered, hopefully you put in your license number, because I'm going to need that when I fill in the form. So that's what I got. So I'll turn it over to Ajay.
Thank you, Pat, very much. So we have some questions in the chat box. How long after cut stump do you think is most optimal for Titi treatment with 50% Gorlon 3A?
How long would it control sprouting?
Yes.
So I think, as I was trying to say, is that really 2 years is a good time to take a look at that. And at that point, you'll know, if you look at mortality, that's a good variable, because it's at that point, it's likely dead. Okay, so it's not going to resprout. And the rest of them are going to continue to grow. Now, the thing about it, though, these herbicides will work for a long time systemically, and particularly, that's very true for imazapyr, to a less extent for radio. So I think that it'll set it back clearly a lot longer than fire, because with fire, you see it within months, it's back. So I hope that answered your question.
So how long after cutting the stump should herbicide be applied? Immediately after cutting?
How long after? Yeah, it's hard to hear. Okay, so yeah, you want to do it right away, honestly, if you can. Yeah, that's pretty important. In many operations, it's hard to find all the stumps. If you're pulling off the top and all that, a lot of those stumps don't get treated. And because of that, they will resprout because they're not treated. And that's been a problem in cut stump treatment and forestry and natural restoration areas, that kind of thing. So it's important to do it within a few hours if you can. And I think Megan went into the treatment very nicely, that you're looking at treating the cambium in particular. The middle of the xylem is not that important. And by doing that, just that green ring, if you would, it's usually brown, just inside the bark, that's the real target that you want it to get there, because you get sprouting from the root collar and you get a little bit of translocation with triclopyr. It is a triclopyr. It is a translocating herbicide. I think that might answer your question.
So Pat, we have another question. Did the inundation of the Flint Rock site impact the effectiveness of any of the treatments?
That's a great question. I really, I was concerned about that, and we went and visited that fall just to check all the tags and see, what it looked like. But I honestly don't think it had that much effect. And there was some difference in the first year. There were, which I attributed to the difference in the hydrology. They weren't huge differences, and honestly, I don't remember exactly what the detail was. And when I revisited the 2-year data, I decided that there was no reason not to average across the 2 sites, because the results really told the same story. That was a concern, that it could have, but I don't think that it really did, except for that first year.
There's a question. Do you think 100% Gorlon would increase mortality over 50%?
Probably. I would bet on it. Doubling the rate, yeah, sure would. Yeah, and it would double the amount of herbicide that you're putting on the site. So we didn't look at that, but my guess is you can. I'm trying to remember. I think the top end of the label actually is if I'm not mistaken, it doesn't go to 100%. I think it's something like 70% or something. I'd have to look at the label to tell you, but I don't think it's full strength with triclopyr, not even with the amine.
But Brian Pelk has a question. Do you think there are chemical differences between habitat and arsenal that account for the pore effect?
Could you repeat the question again?
Hey, Pat, I think I know who was asking. They're trying to determine whether or not the aquatic version of imazapyr had an effect on the pore showing, if the upland version would maybe be similar or not.
Okay, arsenal is a 4-pound, all right? It has no surfactant in it. Habitat is a 2-pound, and because it's aquatic, I'm pretty sure it has no surfactant in it. So there's difference in the amount of the product. So, the rates are going to be quite different. Really, the differences in formulation with the imazapyr products that are out in the market right now, there's a couple generics. There's one that's made in China that I know of, but we didn't test with that. And there's some differences, but generally there's not much difference in the quality of the material, in particular between arsenal and habitat. It's just the amount in the product. Okay, so you would use a different rate. It would be half the rate if you're using arsenal. And I'll add to that, the reason for that arsenal does not have surfactant in it is to improve selectivity. In fact, that was based on work that I did when I was at Arbor. We found that we asked them, please take the surfactant out of the imazapyr because we're going over the top of pines, and we saw that had less phyto when we had no surfactant in the mix. Thanks for the question. But I'll stay for any more questions. I'm going to look for the chat so I can understand what you're saying. There's a chatter in here somewhere. I'll tell you what, Ajay, go ahead and try to read another one for me.
Okay, well, there is only one more question from Eric Brubaker. Eric is a student at Milton and soon will be joining master degree program in Auburn. But anyway, his question is in reference to grasses, so non-target vegetation grasses, were they affected due to overspray of the chemical or because of the, they were like ground active?
Well, again, so yeah, spraying on the grass, obviously that's going to affect that. As I mentioned, though, glyphosate is particularly effective on gramenae for sedges and grasses, etc. The same is true with imazapyr. Now, triclopyr, though, is not as effective, if you would, more tolerant by gramenae, by grasses. So that's a reason that we see more selectivity with the triclopyr, exactly because of that. So it has to be due to the selectivity physiologically because of that. And the effect of overspray is just something you avoid. And we could talk about directed spray, low volume and picking the sites. Things are short. That's really important. Hack and squirt, we do that on shrubs operationally. In the West, it's widely used. They call it hack and daub for shrubs, and it works great, and it's real quick. And again, uses very little chemical. Selectivity, that's the answer to your question.
Are your percentages based on acid equivalent or percent volume of product to water?
Oh, I got that one loud and clear. Yeah, those are a percentage of product, okay? And whenever you do that, you know, talking about percentages, be sure to talk about the particular product, not a percentage of a Mazapure. You know, that really is confusing. Is it the product? Is it the solid? Is it the acid? Is it salt? So, you know, those are rates of the product. And that, I think, is the best way that when you speak to a broad audience, that's the best way to put it for the particular product, because there's so many products for the same product that they have different concentrations. There are different there are products out there that have the same name and different concentrations, which is a real problem and very confusing. So you always look at the label. But I talk about the specific product percentage through this, all of it. Thank you.
The next question is, did you notice differences in treatment effectiveness on different Tai species?
Yeah, the little leaf was actually, there was some of it that was on the TNC site, but we didn't separate that out. And Justin, you know, I think he noted that he thinks that there was some of the little leaf out there. That has a pretty small range. It's just part of Georgia and Florida. That's it, you know. And so, you know, we just grouped them. Yeah.
And is the little leaf the Cliftonia? Because that is leading to Matt Smith's question. Because he said, is research on Cliftonia upcoming? Is there a good reason to suspect it should react similarly?
Okay, what I'm talking about is parvifolia with Cyrilla. Okay, so we didn't address that species. We just looked at the racemiflora, and there may have been some of the parviflora on that site.
But I don't believe we saw much Cliftonia on the site, and that's part of the reason we didn't include it.
Thank you.
Is that correct, Justin, based on your observations?
No, there was not any, well, I say not any. We didn't notice any Cliftonia mixed in with the Cyrilla that we treated. We did look at that at Apalachicola because we thought due to the ranges that there would be more chance of it being there. But there really wasn't. It was primarily swamp Cyrilla or Cyrilla racemiflora that we noticed and what we treated at those two sites.
Well then, Pat, do you think your recommendations could be used on the Cliftonia?
I would say it's likely to be very, very similar. You know, and obviously it's different, but you typically don't see that much of a difference between individual species at the species level. Now at the genus level, yeah. But I think that would, it could be used as a guide, you know, to look at things and refine your rates. You could compare our treatment, maybe the better treatment in your study, and compare some alternates. My suggestion with graduate students is to take a look at rate, always look at rate, look at timing effects. That's something that's very important, particularly of woody plants, and look at long-term results. So make sure you get in a PhD student to finish. No, really, that's a problem with the master's degree, you know, is you need to have some results to write up. But hopefully you work with a group, and a group will have things that go forward through time. Thank you for your question.
Well, all right then. Thank you again, everyone, for joining us today. We hope you enjoyed the seminar. Please contact any of us if you have any more questions. Have a good day and best wishes.
Everybody have a good day. Thank you for your attention.